Good morning, everyone. My name is Krishna Rajarathnam from the University of Texas Medical Branch, UT. Today I'll be talking about molecular mechanisms of protein-GAG interactions. In particular, I'll be talking about a family of proteins called chemokines, which play a very critical role in inflammation and infection, and their function is very much dependent upon how they interact with GAGs, and which is what I'll be focusing the next 40 minutes or so. We have heard a few talks in the last couple of days, and hopefully I'll be able to give you a different perspective. Okay, so let's look at what exactly happens in a disease situation. For example, what I'm showing here is a schematic of the lung on the upper left-hand side, a zoom in of the alveoli, and on the right-hand side I'm showing a cartoon version, an expanded version of what happens, for example, during a bacterial infection. On the lower left-hand corner, what we see is a bacteria, which is sort of green in color. So, for example, if you have bacterial infection in the lung, number of things happen at the transcription level. I will not go into the gory details of all of the immunology, but sort of give you the most relevant aspects of the immune response in the context of what I'm going to be talking about today. So, in the event of a bacterial infection, chemokines are produced at the transcription level. I show the chemokines like these small green circles, and the chemokines which are produced in the tissue and also, for example, by the endothelial cells, go to the cell surface and they bind to the proteoglycans, and in particular to the glycans, and form a concentration gradient. Neutrophils, which are in the circulation, sense these concentration gradients, and the receptors on the neutrophils bind the chemokine. There's a receptor action, cell shape change, and the neutrophils get into the tissue, and once again in the tissue, chemokines direct them to the site of infection. Chemokines actually play two distinct roles. One role is trafficking the neutrophils from the circulation into the tissue, which is what I just described. And the second role is actually killing the bacteria. So we can look at those as something which is holding the gun, but the safety latch is on. And the chemokine actually turn the safety latch off, and the neutrophils start shooting, which in this case is releasing an arsenal of proteases and oxygen radical, which results in killing of the bacteria and in healthy resolution of the inflammation. Okay, so let's look at what happens under disease conditions. Okay, so a dysregulation in chemokine-mediated neutrophil recruitment results in disease, and actually in severe cases can lead to death. So what do I mean by dysregulation in chemokine-mediated recruitment? For example, if the chemokine production is too low, that could result in too few neutrophils, or the neutrophils showing up too late. In other words, the kinetics of the recruitment may not be optimal, resulting in runaway infection, all the way could leading to sepsis. In fact, the children, when they are admitted to ICU, the predominant reason they get admitted in ICU is for the reason they have sepsis. Too many neutrophils could also occur. For example, if the chemokine production is robust and upregulated in which case neutrophils, in addition to killing the bacteria, can also result in collateral healthy tissue damage. In yesterday's talk about Gilani talking about heart disease. And in the case of myocardial infarction, for example, neutrophils are recruited to the site of the tissue damage, and it has been shown that the collateral tissue damage to the heart cells is more detrimental than the heart attack itself. So clearly, if we have an understanding of the molecular mechanisms that regulate recruitment, resulting in healthy resolution, how a dysregulation is cause disease, in principle, not only, of course, leads to a better understanding of the disease process, could also result in targeted therapeutics. Okay, so what is that we know at this time and what is that we don't know in terms of how the infection occurs and the resolution of the infection occurs in the context of neutrophils and chemokines and GAGs. In other words, what are the spatial and temporal dynamics of neutrophils in resolving the inflammation? In reality, we actually know very little of these processes. For example, we do not understand what is the relationship between the severity of the infection or the injury and the amount of chemokine used. If the severity of the infection is high, in other words, there's a lot of bacterial infection, it's a lot of chemokine produced because we want a lot of neutrophils to show up. Okay, and what exactly is the relationship between the severity of the infection and the amount of neutrophils, the kinetics of the neutrophils, and how long the neutrophils are come into the site infection. We really don't know any of this, what one would consider basic information. And as I alluded earlier, we really do not know the molecular mechanisms by which chemokines recruit neutrophils, okay, which I will discuss a bit more in detail down the top. What molecular mechanisms regulate neutrophil recruitment, what molecular mechanisms mediate resolution of inflammation. Of course, all these processes are interrelated, and so we really need to understand to whatever extent we can these processes so that we can design drugs. Okay, so in order to be able to understand the molecular mechanisms of how chemokines mediate trafficking and of course understand the role of the GAGs, we need to integrate the knowledge from different disciplines, okay, so that we can effectively design drugs in a clinical context. Okay, for example, we need to know the chemokine properties, basic structural properties, and that's something which I'll be talking about a bit more in detail. The chemokines exist as both monomers and dimers, so we need to know the structure and the function of both monomers and dimers. And most importantly, we need to be able to test how chemokines do what they do in an appropriate animal model. Obviously, we cannot test humans to find out how the disease process occurs. Okay, the humans are not guinea pigs, so the next best thing we can do is use animal models, and once again, that's what my talk is going to be focused on. So, I'll talk a bit about the protein chemistry and about the animal models. In yesterday's talk, you would have heard fellows from my lab, Dr. Prem Joseph and Krishna Rajarathnam, talk about how structural biology, in other words, the structural basis of the binding process to the GAGs, in particular the geometry, stoichiometry, and so on and so forth, are also critical in understanding the basic mechanisms of how chemokines mediate function. In addition, you also need to know the affinities, the stoichiometry, the kinetics, and the thermodynamics, and things such as how the receptor function and kind of knowledge which I obtained from cellular-based assays, of which I've been not talking about today, but basically focus on the animal models and the chemokine properties. So what I'm going to be talking about in the next few slides are one aspect of chemokine properties, which is their ability to exist as both monomers and dimers. Humans express close to 50 different chemokines, which in addition to recruit neutrophils, they recruit a wide variety of other leukocytes, and they play a wide variety of different functions. Not only in immunology, they play a role in developmental biology, and the fact that all of them exist as both monomers and dimers clearly shows both monomers and dimers play some kind of function in vivo. And so in our context, for example, what exactly do both monomers and dimers play a role in recruiting the neutrophils? Only one of them plays a role in recruiting the neutrophils, and one of them plays a role in activating the neutrophils for tissue damage. So what exactly is the relative ratios of the monomers and dimers? Okay, we need to remember that under healthy conditions, when, for example, you don't have any disease, the level of the chemokine is extremely low, okay? To the extent, if any chemokine is present at all, it's going to be essentially as a monomer. But if you have a bacterial infection, okay, there's under transient conditions, the concentrations could be high enough in the tissue. You could have dimers, or both monomers and dimers, and also in the circulation near the cell surface where it binds to the GAGs, you could have both monomers and dimers. So we need to understand how exactly the monomers and dimers recruit neutrophils. Okay, so how do chemokines mediate function? It turns out the chemokines interact with two different kinds of molecules. They bind to the receptors on neutrophils. The receptors belong to the G protein coupled class, GPCRs, and they also, of course, bind to GAGs. Okay, and they bind to GAGs on the endothelial cells. So the interactions of the monomers and the dimers to the receptors and the GAGs mediate function. So we need to understand all interactions to both the GAGs and the GPRs to be able to understand how these interactions mediate in vivo functions. And okay, so but in today's talk, I'll sort of focus on GAG interactions. So by this time, the last few days, you would have learned quite a bit extensively on GAG, chemistry and structural biology. Okay, so so the chemokines we talk about bind predominantly to heparan sulfate, and which is heparin and heparan sulfate are structurally very similar. And for most of the in vitro studies, we basically use heparin as a surrogate for heparan sulfate. So these GAGs are linear polysaccharides linked to serine of the proteoglycans. Structurally, they have repeating disaccharide units, They're intrinsically heterogeneous because of N-NO sulfation and N-acetylation. And functionally, what we know is the GAG binding is critical for formation of the chemotactic gradient. GAG binding modulates chemokine monomer-dimer equilibrium. GAG binding also protects chemokines from the proteolysis. Okay, these are all from in vitro studies. And so it's it's very likely all three of them are important, though may not. For example, like the chemotactic gradient formation could be more critical than, for example, the proteolysis, but it's very clear that all of them are most probably interrelated in order to effectively do what they do. We know that that the chemokine-GAG interactions are predominantly electrostatic in nature, which should not be surprising considering the fact that GAGs are the most highly negatively charged biomacromolecules, and chemokines are also highly positively charged. And I'm showing the heat schematic of both the monomer and dimer, monomer below and the dimer above, and you can see that the positively charged residues are in blue and the negatively charged residues are in red, and you can see the cluster of blue residues in the dimer and the monomer, and clearly if the interactions are mediated, if it's electrostatic, this means that the interactions are sort of shallow. However high the binding constant is, okay, we heard it's like micromolar, and for the longer GAGs it could be almost nanomolar. The chemokines bind, but they come out, there's a definite on constant and an off constant, which means the chemokines both in the monomer, both the monomers and the dimers in the free and the bound form modulate functions. Okay, this is something which is critical for us to understand. It is just not a GAG-bound chemokine, it's a chemokine both in the free and the bound form. Okay, so how exactly are we going to study the role of the monomers and dimers? Okay, because the one type, which is what we're interested in, okay, is going to exist as both the monomer and dimer, depending upon the equilibrium constant, which is around 10 micromolar. Of course, at 10 micromolar is 50-50, 50% monomers, 50% dimers. At higher concentrations, it's going to be predominantly dimer. At lower concentrations, it's predominantly going to be a monomer. But at any given time, you're going to have both species. And most importantly, in the context of in vivo function, we do not know the concentrations. So we need to understand the function of the monomer in isolation and function of the dimer in isolation. So let's see how this can be accomplished. We know the structure of the dimer. In fact, the IL-8 structure, the chemokine, which I'll be talking about, which is going to be the role model for the rest of my talk, it's a neutrophil activating chemokine. The structure was solved almost 20 years ago, and it exists as a dimer. Okay. And the point I was trying to make was we know the structure of the dimer. So to design a monomer, to make a long story short, because we knew we had a structure, in the past we had introduced a non-natural amino acid to disrupt dimer interactions and trap them off from dimerizing. We have done a structure. In the in vitro functional assays, you see that the activity of the monomer is exactly the same as that of the wild type. 3 different functional assays. If you pay at the concentrations under which you're doing these assays, the highest concentrations is like a micromolar, and the lowest concentration is like a picomolar, and the maximum activity is around nanomolar. And now we know that the wild type dimerizes at micromolar concentrations. So the functional assays in vitro, that's the most important part. In vitro, the wild type does indeed exist as a monomer, so it's not surprising the wild type has the same activity as that of the monomer. But the fact remains, in vivo, as I pointed out, the wild type also could exist as dimer. So what exactly is the activity of the dimer? Once again, those studies were characterized way back in 94, much more recently, like in 2006. We have also characterized the activity of the dimer by trapping the dimer by introducing a disulfide bond at the two-fold symmetry. And we have the structure, and we have shown that the structure of this trap dimer is exactly as that of the wild type dimer, which means that we can be fairly sure that the activity of the trap dimer is the same as that of the wild type dimer. Okay, so how does the dimer activity compare to the wild type? Here I'm showing the binding affinities of a trap dimer and of the wild type. The data for the wild type I'm showing in open circles, and for the trap dimer in closed circles. And the neutrophils, which expresses both CXCR1 and CXCR2, you can see the dimer is less active, but most importantly, it is active. It's about 30-fold less active. For CXCR1, it's about 70-fold less active, and for CXCR2, it's about 20-fold less active. The most important observation is the fact that the dimer is active. Okay, so now we make it clearly a case that that we have well characterized monomers and dimers, and not only that the monomers and dimers have differential activity for the receptors, and also the in vitro studies have shown that they have differential interactions with the GAGs. Okay, and so in the context of in vivo functions, there could be four species in solutions: monomers and dimers in the free form, the monomers and dimers in the bound form. And most importantly, during the course of the recruitment, the relative ratios of the monomers and the dimers in the free and the bound form could be changing constantly, which could be essential for regulating the recruitment, the level of the recruitment, and the duration of the recruitment, and so on and so forth. So to be able to study how this could be happening, clearly it cannot be simulated in an experiment. It has to be done in an animal model experiment. Okay, so let's see how what could be done and what using animal models. Now I'm going to switch gears and talk about how we can use an animal model effectively to gain insights into the molecular mechanisms. So far I was talking about the protein chemistry aspect of it, in other words, the design of the trap monomer and dimer and the function. Now I'm going to switch over to talking about animal models. In particular, we will use the mouse model, which is the most popular animal model. And in particular, we will look at chemokine-mediated trafficking, the regulation, and the neutrophil phenotype. In today's talk, I'll sort of focus only on the trafficking and regulation, but we can also get insights into the phenotype. Okay, so using an animal model, you could infect it, for example, with a bacterium or a virus, and look at what exactly is the consequence of that. So, like in theory, we know that if you have an infection, chemokines are produced, they recruit neutrophils. Depending upon the model you're using, it can kill the bacteria or result in tissue repair, resulting in resolution, a healthy immune resolution, or if there's a dysregulation, tissue injury, and runaway infection. So, but we can also simulate an infection, okay, so which is what exactly we have done. which I sort of show by the red arrow, we simulate an infection by exogenously giving the chemokine. If you administer the chemokine, what happens is the chemokines recruits the neutrophils, okay, but they do not cause any tissue repair because there's no they do not repair any tissue because there was no damage to start with. And of course, they're not going to kill any bacteria because there's no bacteria to kill. So you're able to study in detail a subset of the process, especially being able to answer the question how chemokines recruit neutrophils, and as I'll be showing you in the context of the role of GAGs. So we are going to be using two different models. One model is a lung. We're going to be looking at recruitment of neutrophils in the lung, which I show in panel B. And in panel A, I show the recruitment in the peritoneum, which is both are very well studied animal models, for example, the disease process, and of course, lung diseases are extremely predominant, and role of neutrophils is very established. So in human disease. Okay, so let's cut to the chase and see what happens. Okay, so what we have done is in a mouse model, give different doses of the chemokine, okay, of the dimer, which I show in panel A, of the monomer in panel B, and of the wild type in panel C. Yes, and we are looking at doses of 0.1, 1 and 10 micrograms. So that's a 100-fold range of the dosage. So what exactly are we studying? So we're going to be looking at amount of we're going to sort of try simulate an infection, so which of course results in the chemokine production, and how the amount of chemokine produced. If it's a monomer, what will be the activity, which is the neutrophil recruitment. If it's a dimer, and most importantly, the wild type, it's going to exist on monomer or dimer, and we're going to look at a single time point, which is 6 hours. And we see in panel A at 0.1, there's no activity at all. But at 10 micrograms, as if the floodgates open up, okay, the levels of recruitment are extremely high. Okay, the scale goes from 0 to 40. And in the case of the monomer, recruitment at 0.1 is very minimal, but at 1 microgram is fairly robust. But look at the scale, okay? For the dimer, it goes from 0 to 40, but for the monomer, it's only from 0 to 10. And at a higher dose, you're giving, when you go from 1 to 10, you're giving 10 times more of the chemokine. And the neutrophil recruitment is actually lower, okay, which you would have never expected. But I've always thought more chemokine, more neutrophils, but that's not what it is. And in the case of wild type, okay, it doesn't look like the recruitment profile of the dimer or the monomer. It's actually in between. And at 10 microgram shows the highest. 1 microgram you see some recruitment. Okay, so you make quite a few observations which we could have never predicted from our in vitro studies. Okay, the most important observation was that the high levels of the recruitment of the dimer, if you remember, that activity of the dimer for the receptor was, if anything, lower, okay, which says by default that the higher recruitment must be due to GAG interactions. And once again, the GAG interactions doesn't involve any signaling, okay. As far as we know, it's involved in formation of a gradient, okay. So this gradient formation clearly influences the recruitment, most importantly, the kinetics of the recruitment. Okay, that's what we think is happening. I'm showing the same data as what I showed in the previous slide, but plotting it in a different way so that we get a better appreciation of the differences in the recruitment levels between the wild-type monomers and dimers. In panel A, we are looking at the recruitment at 0.1 microgram. We can clearly see the monomers active, dimer is not active, but the wild type is somewhat active. The most critical aspect is the scale, 0 to 0.3. At 1 microgram, the monomer is active, dimer is inactive, and the scale is up to 10, 0 to 10, about 30 times more recruitment. And in panel C, we are looking at the data for the 10 microgram data. Clearly see the dimer is more active compared to the wild type and the monomer. And what is most interesting, as I alluded earlier, the monomer activity actually comes down compared to the data, the 1 microgram data. So far we were looking at data at a single time point. It's a snapshot. It was a snapshot at 6 hours. What happens at other time points? Do we see the same trend, or the trends could change? On this panel A summarizes all of the data for the wild type monomer and dimer at 4 different time points, 2, 6, 12, and 18. Okay, panel B shows the profiles of each of the variants, the dimer for panel B, monomer for panel C, wild type for panel D. And what we see is that at 2 hours the recruitment is really low, and at 6 hours it peaks, and at 12 and 8 it drops very sharply. It's sort of a skewed bell-shaped curve. In panel C, the recruitment for the monomer at 12 hours it slightly goes up, but the recruitment levels remain low. Okay, in this case, I'm plotting all of the data going from 0 to 40, and the data is 10 micrograms. In the case of the wild type, the recruitment levels at 12 hours is as high as 6, indicating that the fact that the wild type can exist as both monomers and dimers results in, I would say, robust, sustained recruitment. Okay, so for the recruitment to occur, obviously the IL-8 has to come from the tissue from where it is produced into the blood, and clearly the rate at which the IL-8 is produced will influence the reverse kinetics and duration of neutrophil trafficking. And what we observe is that IL-8 is rapidly mobilized to the bloodstream. I mean, we start seeing the IL-8 even within 15 minutes. So its kinetics is not the rate-limiting step. It's the neutrophil recruitment, the trafficking, which is a rate-limiting step, which is not surprising considering neutrophils are much bigger and they have to really squeeze in through all these different barriers compared to the chemokine, which is much smaller. And the monomer-dimer equilibrium itself doesn't really seem to influence much of the chemokine trafficking, because the recruitment of the dimer is sort of similar to that of the wild type. And what is interesting is also that the I monomer levels in the serum, okay, which you can see as outermost data indicates that the binding to the it binds, it's in vivo binding the GAGs is weaker. So the neutrophils show up like clearly, and as I said, there are two parts to the chemokine-mediated neutrophil recruitment. One is the recruitment itself, and one is the neutrophil action. And what I'm showing here is that in panel A is the control when there's we just give PBS, no chemokine. Panel B is IL-8 mediated neutrophils in the tissue, and panel C is actually a viral infected tissue. And the B looks very much A, showing that the neutrophils show up, okay, but they really don't cause any damage. So which allows us to study the process of the trafficking. And it's actually, if you think about it, it's not surprising because there are no chemokines in the tissue. All of the chemokines have already gone into circulation. So neutrophils are just waiting there for some kind of a signal for them to start causing, start releasing the proteases, for example, like in the case of a bacterial infection. In panel D shows the protein levels in the lung, and you see there isn't much protein, and definitely not monomer-dimer in the wild type, suggesting once again that the integrity is not compromised. Okay, so far I've been talking to you about the lung. So the lung is just one of the many different organs we have. So what applies for lung, does it apply to other organs? So we have looked into the peritoneum, and what you see is that once again we look at this dose dependence, looking at neutrophil levels of the wild type monomer-dimer at 4 different doses, 0.1, 0.25, 1, and 10. Now the activity of the wild type is much higher, okay, even at like the lowest dose. Okay, wild type has much higher activity than the monomer, which was not what we observed for the lung. Slide sort of summarizes the neutrophil levels for the different IL-8 variants, and you see especially for the monomer, the recruitment between like 0.1 and 10 is very similar, indicating there's no correlation between chemokine produced and neutrophil recruitment. Okay, the activity of the dimer is much lower, and the wild type shows much higher activity. So clearly indicating there's a synergy between the monomer and dimer interactions, which facilitates neutrophil. And this data sort of summarizes the recruitment profiles in peritoneum versus lung. And we see like at low doses the dimer is inactive, and but at high doses it's substantially more active in the lung. And both the monomer and dimer show this sort of inverted U shape profile. In other words, lower activity at higher doses, which is more profound for the lung. And once again, the largest difference is in the wild type, which is what matters. The wild type is what exists in nature, and the wild type is definitely more active at low doses. Okay, so that is the profile, the recruitment profile is sort of shallow, whereas in the case of the lung it's more profound, clearly indicating tissue architecture, and most probably the tissue-specific differences, the GAG production, the GAG composition also plays a role. So far, we have seen that there are differences in the recruitment between the monomer and the dimer and the wild type. And I sort of indicate that can't be because of the receptor interactions. It must be due to GAG interactions. But now I'm going to show you data that it is indeed directly from the GAG interactions. I'm showing a schematic of IL-8, and residues which are important for GAG binding are shown in red. And these we know from the in vitro studies, and you can see they cluster on one phase of the molecule on the helix on the 310 loop right adjacent to the helix. So what we did was to create single mutations of all these amino acids on the wild type background, and we looked at the recruitment of the mutants and compared to the wild type. So we did experiments at 3 different doses, 0.25, 1, and 10. And we can see right away that the recruitment of the mutants were lower. We know that at the 0.25 micrograms, the wild type exists predominantly as a monomer, and at 10 microgram, it exists predominantly as a dimer. And so the lower binding of the monomer and the dimer results in reduced recruitment, clearly establishing a causal relationship between GAG binding and neutrophil recruitment. Okay, so far we have been looking at neutrophil trafficking profiles of one single chemokine, that of interleukin-8 or CXCL8. But there are actually 7 different chemokines which recruit neutrophils. So the question is, do all other chemokines recruit like IL-8, or there are chemokine-specific differences in the recruitment? So what we have done, in addition to looking at the recruitment of IL-8, we also looked at the recruitment of MGSA, also known as CXCL1, and I'm showing you the data here on the right-hand side panel. Shows the recruitment of the wild type, the monomer and dimer at the 3 different doses. On the left hand, you can see the recruitment at the individual concentrations, 0.1, 1, and 10. And we can see right away the wild type is highly active, more active than the monomer or the dimer, showing that the monomer-dimer synergistically enhance recruitment. And what is interesting is that the 1 microgram data, the recruitment of the monomer is actually lower. It sort of was higher for IL-8. We can look at the comparison figure very soon. And the recruitment at the 10 microgram data is almost like IL-8. So even at individual doses, there are differences and there are also commonalities. But there's the overall, the profiles are sort of similar compared to IL-8. Okay, so in this schematic, we can clearly compare and contrast the recruitment profiles of IL-8 and CXCL1. You can see that at the lower doses, 0.1 microgram data, the profiles are sort of similar, but IL-8 is more active, but definitely the wild type is more active at this dose for both the chemokines. And at 1 microgram data, the monomer is highly active for IL-8, but not for MGSA. And at 10 microgram data, the recruitment profiles are very similar. And the differences because of GAG interactions, because both IL-8 and MGSA bind to the receptors in a very similar fashion. And so in other words, there are chemokine-specific differences. But the overall observation that the wild type is more active than the monomer or dimer remains the same for both the chemokines. So far we were looking at the recruitment in the peritoneum. So what about in the lung? So now I'm showing the recruitment of GSA in the lung model at the 1 microgram and at the 10 microgram. And at the 1 microgram, the recruitment of the wild type is fairly high, and the activity of the dimer is fairly low, and the monomer is sort of in between. At the 10 microgram, actually, the recruitment is similar to what we saw in the peritoneum. And so in the next slide, let's look at how the recruitment compares between MGSA and IL-8 in the lung model. Okay, so on panel A, we're showing the data for IL-8, and in panel B, we're showing the data for CXCL1. And in panel A, we show the data at 0.1 microgram day, for the 0.1 microgram, which we did not do for MGSA. But we can clearly see that in the lung, okay, the monomer is fairly active for MGSA, but whereas it is not for IL-8, and also that the monomer is more active for IL-8, whereas the wild type is more active for MGSA. So whereas we saw that the profiles were sort of similar in the peritoneum, there are distinct differences in the recruitment profiles in the lung, indicating that once again the differences must be due to GAG interactions, and most likely due to the differences in the gradient formation, which is a consequence of the tissue architecture and most likely the GAG, nature of the GAG distribution. Okay, so I was sort of talking about how using animal models, we can get mechanistic insights at a molecular level of how the chemokines, in particular the monomers and the dimers, via the GAG interactions mediate the recruitment. We definitely saw the levels of recruitment, the kinetics of the recruitment. All of them are very much intimately connected to the recruitment process. Of course, these are absolutely essential for a healthy resolution. In addition to that, we do know from other studies that during the recruitment, proteases, MMPs, release GAGs, okay, which means they're not only immobilized GAGs, they're also free GAGs, okay, as I've shown in the schematic. And these studies have shown that the chemokines bound to the free GAGs also regulate the recruitment process. So clearly we have the monomers and dimers in solutions, the monomers and dimers bound to immobilized GAGs, and it seems monomers and dimers bound to free GAGs, altogether regulating the recruitment process. So this is where things stand. So let's summarize. Okay, so in summary, clearly an aspect of the molecular mechanisms, in this case the chemokine monomer-dimer equilibrium, we show dynamically regulates neutrophil recruitment. The word dynamic is critical because it is not a constant. The monomers and the dimers are constantly changing. I mean the levels, that's because the protein concentrations are constantly changing as a function of space and time. And so their interactions with the GAGs and the GPCRs that regulates the recruitment. And we also show that the differences in GAG interactions between monomer and GAG interactions to the dimer result in differences in gradient formation and hence in the recruitment. And we also show that tissue-specific differences, in our case, we showed data for the lung and the peritoneum, is because of the GAG interactions, resulting in differences in the recruitment. And we also show that there are chemokine-specific differences, and it also must be due to differences in GAG interactions, not definitely the receptor interactions, because both the chemokines bind to the receptor in a very similar fashion. And most importantly, we also show that between the monomers and dimers, the dimer-GAG interactions are more critical, and so disrupting dimer-GAG interactions could result in clinically relevant therapeutics. So let me stop here and thank you.